It is October 2015. A genetics laboratory at the University of Padua in the flat farmland west of Venice. Inside, a technician positions a tray of amplified DNA fragments into a next-generation sequencer, the same class of machine that commercial ancestry companies use to tell paying customers where their grandparents came from. But these fragments did not come from a living person. They did not come from a cheek swab or a vial of saliva. They came from dust. Microscopic particles, vacuumed decades earlier from the surface of a cloth that has been argued over for more than 600 years. A cloth kept in a climate-controlled reliquary inside a chapel in Turin, Italy, behind bulletproof glass and under the custodial authority of the Vatican. The cloth is 4.4 meters of hand-woven linen, a single continuous piece, threaded in a 3-to-1 herringbone twill. It bears the faint, straw-yellow outline of a man. And depending on who you ask, it is the most rigorously studied artifact in human history, the most elaborate hoax ever produced, or the burial shroud of Jesus Christ. What the sequencer is about to return will not settle that argument. But it will make the argument much harder to have. Because the DNA on this cloth does not tell a simple story. It tells several stories at once, layered on top of each other like sediment, and at least two of them should not be possible in the same object. Let me walk you through what they found. For most of the 20th century, the scientific conversation around the Shroud of Turin had a clear architecture. 2 camps, 2 pieces of evidence, and a clean divide between them. Believers pointed to the image. The image is, by any honest account, genuinely strange. In 1978, a team of roughly 30 American scientists, known collectively as STURP, the Shroud of Turin Research Project, spent 120 continuous hours examining the cloth in the royal palace adjoining Turin Cathedral. X-ray fluorescence, ultraviolet photography, infrared spectroscopy, microscopes powerful enough to resolve individual fibers, every instrument they could carry through the door. Their final report, published in 1981, contained a sentence that has haunted forgery theorists ever since. The body image is that of a real human form of a scourged, crucified man. It is not the product of an artist. So what is it? Not paint, not dye, not ink, not any applied substance they could detect. The discoloration exists only on the outermost layer of each linen fiber, a yellowing so shallow it is measured in fractions of a micrometer. Darker areas are simply zones where more individual fibers have been discolored, like a halftone photograph printed by a process no one can name. That finding did not have to mean anything super, but it meant the cloth had properties that resisted easy explanation. Any painting, rubbing, or scorch should have penetrated deeper into the threads. This image did not. Then came 1988. And the date. On the morning of April 21st, 1988, in the sacristy of Turin Cathedral, the shroud was unrolled and laid flat before representatives of 3 laboratories: Arizona, Oxford, Zurich, the most respected radiocarbon facilities in the world. A textile expert and 2 university professors argued for nearly 2 hours over where to cut the sample. In the end, a single narrow strip was removed from the lower left corner of the cloth. It was divided into portions, placed in coated containers, and sent to each lab along with 3 control samples of known age. The result, published in Nature in February 1989, was emphatic. All 3 labs placed the linen in a window of 1260 to 1390 AD. The statistical analysis said the data were consistent. The conclusion said the shroud was medieval, not 1st century, not ancient, a product of the Middle Ages. The headline wrote itself: "Case closed." Except it was not closed, not even at the time. Because the sample came from a single location, the outer edge, the part of the cloth that had been gripped, folded, kissed, and handled more than any other section across centuries of public exhibition. This was the corner that nuns had stitched after the fire of 1532. The corner most exposed to candle smoke, incense residue, and centuries of atmospheric carbon. It was, in short, the worst possible place on the entire shroud to take a representative sample. Radiocarbon dating measures carbon-14 content. It dates the material, not the event. If newer carbon has been added to the surface by contamination, biological films, or restoration, the result skews younger. And there was no chemical analysis performed on the removed sample before it was dated. It was cut, distributed, and measured. The original protocol had called for chemical characterization. That step was skipped. It took 30 years and a lawsuit for the underlying data problem to surface in print. In 2019, a team led by French researcher Tristan Casabianca obtained something that neither the Vatican nor the British Museum had been willing to release for 3 decades: the raw measurement data from the 1988 dating. Casabianca's team had to file a legal request with the University of Oxford. They won. What they found, published in Archaeometry, Oxford University's own journal of archaeological science, was not subtle. The 12 individual subsample measurements were statistically heterogeneous. They failed the Ward and Wilson chi-squared test, the same statistical procedure cited in the original Nature paper, to justify combining the results into a single age estimate. And certain raw values had been altered before publication, using a methodology that was never stated. The published result in Nature was a polished average. The raw data underneath it were a mess. This does not mean the shroud is ancient. It means the one scientific result that most educated people cite when they say the question is settled does not, by its own statistical standards, meet the threshold for reliability. The numbers do not cohere. They never did. Into that gap, something walked in that nobody expected. DNA. Gianni Barcaccia is a plant and animal geneticist at the University of Padua, not a theologian, not a member of any syndenology society. His research background is in crop genomics and biodiversity. When he and his team, which included mitochondrial DNA specialists Alessandro Achilli and Antonio Torroni from the universities of Perugia and Pavia, obtained access to the dust particles that had been vacuumed from the shroud during STIRP examinations in 1978 and again during the radiocarbon sampling in 1988, their goal was forensic, not devotional. They wanted to know what biological material existed on the cloth and whether it could tell a geographical story. The dust came from 5 separate filter samples. 4 corresponded to areas of the body image, face, hands, gluteae, and feet. The 5th came from the lateral edge, the same corner used for radiocarbon dating. Using standard protocols, they amplified plant chloroplast DNA and human mitochondrial DNA, sequenced more than 200 fragments, and began mapping what they found to known species and known human genetic lineages. The plant DNA alone was a globe-spanning inventory. 19 separate taxa, Mediterranean clovers, ryegrasses, and plantains you would find in any southern European field, European spruce trees, North American black locust, and then the puzzles, East Asian pear and plum varieties, species whose primary center of origin is in China. Some of these species could not have been introduced to Europe before the voyages of Marco Polo or Christopher Columbus. Whatever this cloth is, it has been in sustained contact with the botanical output of at least 4 continents, across a time span that stretches well beyond the medieval period. But the human DNA is where the floor drops out. Among 93 mitochondrial DNA amplicons, the team identified sequences belonging to haplogroups scattered across the human family tree. H1 and H3, extremely common in Western Europe and peaking among Iberian populations at roughly 25% frequency. H13 and H33, concentrated in the Near East, around Anatolia and the Caucasus. R0A, a lineage typical of the Arabian Peninsula. L3C, a haplogroup so rare it appears almost exclusively in East Africa. And then there was a cluster that did not fit any European or Near Eastern model. Haplogroups M39, M56, R7, and R8. All four are characteristic of the Indian subcontinent. R8, specifically, is found essentially only in eastern India. These are not haplogroups that migrate casually into Western European populations. They are geographically anchored, regionally specific, and deeply unusual to find on any European artifact of any age. What made Barcaccia lean forward was the degradation pattern. In ancient DNA analysis, fragment length is a rough proxy for age. DNA breaks down over time. Shorter, more degraded fragments generally indicate older biological material. The most degraded human DNA sequences on the Shroud, the fragments that were most broken down, were not European. They were Indian, and they were not confined to the outer edge. They appeared across the interior body image areas of the cloth, the regions that had been more protected from environmental contamination and less exposed to handling. Barcaccia's team also flagged another detail that tends to get lost in the coverage. One of the most abundant human mtDNA haplotypes detected on the Shroud matched lineages typical of the Druze community, an ethnic group with roots in Egypt that today lives primarily in restricted areas across Syria, Jordan, Lebanon, Israel, and Palestine. The Druze haplotype was not peripheral noise. It was among the strongest genetic signals on the cloth. If you are building a channel around the moments where DNA evidence quietly overturns historical assumptions, the Druze haplotype on the Shroud of Turin is exactly the kind of detail that rewrites the map before anyone notices. We are covering more stories where genetics and geography collide, and if that is the kind of evidence you want to follow, the subscribe button is below. Barcaccia's interpretation in his 2015 paper, published in Scientific Reports, was careful but pointed. The presence of Indian origin haplogroups across the body image areas of the cloth, the protected interior that saw less external contact, suggested something about the fabric itself, not merely about the people who touched it in later centuries. His team noted a linguistic detail that scholars had observed before but never connected to genomic data. The word Sindan, the Greek term used in the Gospel of Mark to describe Jesus' burial cloth, may derive from Sindhya or Scythian, an old term for fabric imported from India. First-century trade routes between the Indian subcontinent and the eastern Mediterranean were vigorous and well-documented. Pepper, textiles, gemstones, and spices flowed west through the Red Sea ports. High-quality linen could have been manufactured in India, shipped to Alexandria or Caesarea, and purchased by a wealthy family in Jerusalem for a burial. Now lay that over the Shroud's claimed historical path, Jerusalem after the crucifixion, then possibly Edessa, in modern southeastern Turkey, where early Christian tradition places the image of Edessa, a cloth bearing a miraculous face. Then Constantinople, where Crusaders sacked the city in 1204 and took relics by the shipload. Then France, where it surfaced in the hands of a knight named Geoffrey de Charny in the 1350s. Then the House of Savoy, which moved it to Turin in 1578. Indian linen, Middle Eastern blood signatures, Druze lineage mitochondrial DNA, East African traces, Western European haplogroups consistent with centuries of veneration in France and Italy. A marine worm DNA sequence from the North Pacific, near Canada, that defies any simple geographical narrative. That is not the DNA profile of a medieval European forgery. A forger in 14th century France would not have had access to linen-carrying Eastern Indian mitochondrial material in its oldest degradation layers. They would not have planted Druze community haplotypes that no one could identify until the 21st century, and they would not have distributed those traces across the interior of the cloth, under the body image, where contamination from public displays would be least likely to reach. But, and this is the turn the evidence demands, it is also not proof that the cloth is genuine. Hugh Ferry edits the newsletter of the British Society for the Turin Shroud. He is one of the more intellectually honest voices in this debate because he holds no fixed position. He has said publicly that he is roughly 40% convinced the shroud is authentic and 60% inclined toward it being a forgery. He considers both sides and changes his estimates as new evidence arrives. On the DNA findings, Ferry and other cautious voices raise a structural objection that is hard to dismiss. Distinguishing between DNA deposited during manufacture and DNA deposited during later contact is, with current methods, impossible. Fragment degradation provides a rough signal, but it is not a reliable clock. A shorter fragment might be older, or it might simply have degraded faster because it was exposed to more heat, more humidity, more enzymatic activity from microbial colonization on the cloth's surface. The contamination problem is symmetrical. It weakens the 1988 radiocarbon date by introducing newer carbon, but it also weakens any attempt to trace the cloth's origin through surface DNA. Centuries of veneration mean centuries of skin cells, breath moisture, and handling by thousands of people whose identities we will never know. Each left behind a genetic signature. Barcaccia himself acknowledged this tension. His published conclusion was deliberately open-ended. The DNA diversity, he wrote, does not exclude a medieval origin in Europe, but it would also be compatible with the historical path followed by the Turin Shroud during its presumed journey from the Near East. What neither side of this debate can honestly do is dismiss the other. The Indian signal is there. The Druze haplotype is there. The haplogroup diversity spanning 3 continents is there. The question is whether those traces were laid down during the cloth's creation or accumulated across centuries of transit and veneration. And that question, right now, has no definitive answer. There is one more complication, and it pulls the timeline in yet another direction. In 2022, Liberato De Caro, a researcher at Italy's Institute of Crystallography, part of the National Research Council, published a study using a completely different dating approach. Wide-angle X-ray scattering, or WAXS, measures the structural degradation of cellulose fibers over time. As linen ages, the crystalline structure of its cellulose breaks down in measurable ways. De Caro tested a thread from near the radiocarbon sampling area, a fragment approximately 0.5 millimeter by 1 millimeter, and compared its degradation profile against a calibration set of reference linens with known ages spanning from 3000 BC to 2000 AD. The shroud's profile matched most closely with a piece of fabric from the siege of Masada in Israel. That linen is independently dated to 55 through 74 AD, 2,000 years old, squarely within the lifetime of Jesus of Nazareth. The headlines were immediate, but so was the criticism. The wax dating method was developed by the same team that applied it to the shroud. It has not been independently replicated by other laboratories using other samples. Cellulose degradation depends heavily on storage conditions, temperature, humidity, light exposure, and microbial activity, all of which are impossible to reconstruct with precision over 2 millennia. A cloth stored in the dry heat of Masada's desert fortress would degrade differently from a cloth that spent centuries in the damp churches of France and the variable climate of northern Italy. Critics, writing in Skeptic magazine, further noted that key members of the research team are committed authenticists. At least 2 are followers of the Italian mystic Maria Valtorta. One co-author, Giulio Fanti, has publicly stated his belief that the shroud is genuine. The journal in which the study appeared, Heritage, is a legitimate but relatively new MDPI publication, not a top-tier archaeological science venue. De Caro himself has acknowledged these limitations and explicitly called for independent laboratories to repeat the wax analysis on additional shroud samples. That is the correct scientific response. But the Vatican, which has owned the shroud since the death of King Umberto II in 1983, has shown no willingness to authorize new sampling campaigns. and so the field sits in an uncomfortable equilibrium. The one dating result the world accepted is statistically compromised. The new result that challenges it has not been replicated. The DNA evidence supports neither a simple medieval nor a simple ancient origin. Every method that has been applied to this cloth has produced results that are either contested, incomplete, or mutually contradictory. The shroud does not cooperate with certainty. It never has. What I keep returning to, after reading Barcaccia's paper and Casabianca's statistical reanalysis and De Caro's X-ray data and Ferre's measured skepticism, is not the question that most people ask. Most people want to know, is it real? Did this cloth wrap the body of Jesus? That question may be permanently unanswerable. There is no reference genome for a first-century Galilean carpenter. There is no chain of custody stretching back 2,000 years. And even if every dating method converged on the 1st century, that would still not prove whose body made the image. But the DNA evidence does something that none of the previous investigations managed. It proves, beyond reasonable dispute, that this object has moved through the world in a way that no simple narrative covers. Indian genetic signatures in the most degraded layers. Druze community haplotypes concentrated in the body image regions. East African mitochondrial sequences that have no business being on a piece of European medieval cloth. Plant species from China and the Americas woven into the same dust record as Mediterranean field grasses. This is a cloth that has been breathed on by civilizations. And that matters whether the linen is 2,000 years old or 700. Because if it is medieval, then the forger produced an image that 30 American scientists with 20th century instruments could not explain, used a cloth already carrying Indian origin DNA in its deepest degradation layers, decorated it with bloodstains containing markers that modern forensic pathologists associate with severe trauma, and anticipated analytical techniques that would not exist for 6 centuries. That is not a forger. That is something else entirely. And if it is ancient, then we are holding a textile whose genetic passenger manifest mirrors the trade routes, migrations, and demographic movements of the 1st and 2nd millennia, with a precision that no scholar could have reconstructed before the age of genomic sequencing. Either way, the shroud is not what the 20th century decided it was. Right now, today, there are geneticists in Padua, and crystallographers in Bari, and statisticians in France, and textile historians in the United Kingdom who all want the same thing. New samples. Taken from the center of the cloth, not the edge. Tested by multiple independent laboratories. Using protocols designed for degraded ancient DNA, not 1980s-era radiocarbon techniques. Until that day, the shroud remains exactly what its DNA says it is. A record of contact, a ledger of touch, stretching from eastern India to western Europe, from the Arabian Peninsula to East Africa, written in the only language that does not lie and does not fade completely. The base pairs of every person who ever touched it, venerated it, repaired it, studied it, or simply stood close enough for a skin cell to drift down onto its surface. And somewhere in that record, possibly layered beneath centuries of accumulated genetic noise, sits the trace of the person whose image still gazes out from the linen. Unsequenced, unnamed, present in the data, but invisible to our tools. The Druze populations of the Levant still carry haplogroup H33. The communities of eastern India still carry R8. The descendants of those genetic lineages are alive right now, walking through markets and riding buses and checking their phones, carrying in their mitochondria the same sequences that a machine in Padua pulled from 2,000-year-old dust. The past is not behind glass in Turin. It is circulating, in blood.